Hello and welcome to our research output presentation on the bioprospecting of endophytes and their potential agricultural application. This session will be presented by me, Anis Nadia, and my partner, Muhammad Nur Abadi. We are representatives from Universiti Brunei Darussalam's Department of Environmental Life Sciences. This project is conducted under the supervision of Dr. Pooja Shivanand and Dr. Faizah Matali. Without further delay, let's begin. By 2050, the United Nations expects the global population to reach 9.7 billion, and the agricultural practices are under pressure to provide increased yields to feed everyone. Additionally, due to global warming and the climate abnormalities associated with it, crops typically encounter increasing abiotic and biotic stresses, which severely affects their growth and productivity. However, the current practice of intensive agriculture, which is heavily reliant on the use of chemical fertilizers, herbicides, and insecticides to address global food challenges, is not only environmentally harmful, but also not economically feasible. There is a need to apply sustainable practices to problems related to agriculture and food production. Therefore, we suggest the bioprospecting of endophytes with agronomic potential as a sustainable alternative to increase crop yield. So, what are endophytes and what are their significance? Endophytes are microbes such as fungi and bacteria that are known to colonize internal plant tissues of various plant parts, such as roots, leaves, fruits, and even seeds, without causing any harm to the host plant. Instead, they form a symbiotic association with their host plants. For instance, by providing a defense mechanism against pathogens in return for a nutrient-rich living condition. Compared to rhizosphere or free-living microbes, endophytes are better protected from environmental factors such as extreme fluctuations in temperature, pH, and water availability, as well as having to compete for food or nutrients. Many kinds of endophytes, by living inside plants, bring out the hidden strengths of their host, such as their growth potential or resistance to disease and harmful insects. Hence, endophytes can be used as a valuable tool in agriculture for improving crop productivity. Over the recent years, there have been a steady increase in research on the application of endophytes in agriculture. Common findings were mainly on endophytes as a plant growth promoter and as a biological control agent. Endophytes were reported to directly promote plant growth by producing phytohormones such as indole acetic acid and enzymes, as well as indirectly by contributing to plant nutrient uptake through nitrogen fixation, phosphate solubilization, or iron transformation. One study has successfully enhanced growth of rice seedlings that have been inoculated with two species of bacterial endophytes that has been isolated from the mangrove. This was concluded due to the presence of high hydrolytic enzyme activity and also due to the ability of the isolates to synthesize plant hormone IAA and to solubilize phosphate. In regards to the application of endophytes as biocontrol agents, endophytic bacteria in particular enhances the host plant growth by discouraging the growth of phytopathogens and plant pests. They can produce substances that can antagonize phytopathogens like antimicrobial compounds, toxins, siderophores, and hydrolytic enzymes. Both bacterial and fungal pathogens can be targeted by endophytic bacteria. Multiple studies regarding endophytic bacteria have been demonstrated to successfully suppress fungal diseases in crops such as black pepper, potato, and wheat. Additionally, in 2019, endophytic bacteria Pentoia vagans was commercialized as a biocontrol agent for fire blight, that is a bacterial disease affecting fruit plantations such as apples and pears. This finding had shown that the production of antibacterial metabolites by the endophytic bacteria itself contributed to its biocontrol activity. Furthermore, plant roots offer a perfect environment for the degradation of heavy metal by microbes, thereby reducing toxic effects on contaminated sites. Endophytic bacteria, in association with plants, efficiently promotes the remediation of contaminated sites as they possess traits that can remove contaminants, such as production of iron chelators, siderophores, organic acids, and various degrading enzymes. Combined effects of all these influences the contaminant availability for uptake and degradation of pollutants. This type of mechanism is more effective compared to bioremediation, where the microbes may not be able to survive at a site or degrade a pollutant because it may not be bioavailable. A study in 2015 reported that endophytic bacteria Bacillus pumilus significantly increases metal uptake from contaminated soil into tissues of plant, Sedum plumbisicicola, while simultaneously promoting plant root growth and shoot length uptake. This is proof that using plant endophyte association for removing heavy metals can be a promising green technology. As for endophytic research being conducted within our Department of Environmental Life Science in UBD, currently we are focusing on the bioprospecting of endophytes, specifically within mangrove forests. Recently, we managed to isolate a total of 35 endophytic bacteria from the leaf and bark of two mangrove plant species, namely Rhizophora apiculata and Nipah fruticans. These isolates were found to be mainly gram-positive endospore-forming bacteria. At the moment, DNA extraction of isolates are being carried out, which will be then sent out for identification. You might wonder why we are focusing on the bioprospecting of endophytes within mangrove. Well, this is because mangroves are one of the several forests which can be found within Brunei. The mangrove ecosystem is characterized by periodic tidal flooding, which causes factors such as salinity, temperature, and nutrient availability to be highly unpredictable. Hence, the ecosystem is known to harbor high microbial biodiversity. The unique and extreme habitat are likely to confer endophytes with special physiological adaptations. Additionally, mangrove plants are mostly known to have high medicinal values. Therefore, mangroves endophyte have a high potential of producing novel bioactive compounds.
Next is the result of our enzymatic screening where we selected 5 isolates from each plant, Nipah fruticans and Rhizophora apiculata. From table 2 here, we can see that we got 8 isolates that produce cellulase, 7 isolates produce amylase, and 3 isolates produce protease, while 2 isolates produce lipase. As you can see from table 2, we use enzymatic index where the halozone diameter divided by the isolate's diameter. Any enzymatic index more than 1 would be positive presence of the enzyme. This number also can show which isolates produce more enzymes compared to each other. Here are the figures where the enzyme has been identified by screening using respective substrates agar. So why we study this enzyme specifically from endophytes? These enzymes are very useful in agriculture. First of all, cellulose. Cellulose helps to enhance the crop growth by breaking down some of the cellulose bond inside the plant cell so that the cell can enlarge in size. It is a control agent of the plant diseases, where the combination of cellulase, hemicellulase, and pectinase are broadly applied. Certain fungal cellulase are able to degrade cell wall of the plant pathogens. It is evident that the cellulitic microorganisms are participating in many processes in assisting the plant, such as rhizosphere soil decomposition, increasing the availability of nutrients for the plant, controlling plant pathogens, facilitating root colonization, and penetration of cereal crops, improving yield and nutritional contents. Next, we have amylase and protease. Amylase and protease can be a utilizer of agro-industrial waste, such as fodder of poultry, where it contains a very rigid keratin structure, accounts for 5% of the body weight, and is a rich source of proteins for feed and food. Poultry waste can be degraded into feed and food by the keratinolytic process. Waste such as paddy hulls can be broken down into sugars by amylase. Brunei Darussalam aimed to achieve at least 10% of energy generation from renewable resources by year 2035 to reduce the impact of climate change and reliance on fossil fuel reserve. Brunei, with its vast number of rainforests and rising agricultural sector, should take advantage of utilizing biomass solid waste as an alternative energy source. Here are the next potential research features for endophytes. First, the identification of potential plant growth promoting isolated endophytic bacteria, which are my master's project. Second, the identification of nitrogen-fixing endophytes. Third, the identification of phytochemical with insecticidal constituent from endophytes. Fourth, the seed quality enhancement with endophytes. Fifth, the trimorgenic effect of large animal towards secondary metabolites of endophytes. And the last one, mangrove endophytic bacteria promotes deforestation growth. Endophytes are naturally occurring biocontrol agents with potential beneficial uses in the plant. The use of endophytic microorganisms for the plant disease control is relatively new and unexplored area of research. Little is known about the interaction between the endophytes, host plant, and the pathogen. However, further research is necessary to advance the science for practical uses. There is a distinct lack of knowledge of any limitation, especially in demonstration of field efficacy. The use of endophytes may be limited until we learn a better understanding of ecology of endophytes. It is unclear as to what factor, if any, within endophytes or host plant detect specific host colonization potential, raising some questions. And here are our references. I would like to say thank you to everyone who are involved in this project. Here are our contact info. Thank you for watching this video.